Okay. So let's have a class. Our first lecture, exam 1, lecture 1, we will talk about the introduction and history of the microbiology. And I also want to say, some of you already living in a microbiology world, and there was some material or similar or the same, but will be more depth here. Because I know there is about 10 students who is taking my previous classes, certainly helps, but majority of you have been taking yet. Okay, so lecture 1 we'll talk about that. So microbiology. So this is microbiology. So what are we going to talk about? Microbiology, we said very simple. This is science of microorganisms. Now, what we're going to learn in this class, we already see the syllabi will be 4 exams. Exam 1, we will talk about the basic knowledge of microorganism, including characteristics, staining technology, enumeration, classification. That's very basic. Exam 2, I think this is lots of you are interested, especially in the future you go to the medical school. We will talk about this infectious disease related to microorganism. And basically, we're going to focus on the bacteria here. Okay, that's exam 2. Exam 3, this is one kind of challenge for most of the students, but some of you are really good. We are going to talk about biochemistry. Biochemistry, we will talk about is basic, the metabolic cycle. And we also talk about the genomics, we talk about DNA replication, transcription, and translation. If you have already taken biochemistry class, certainly it helps. And if you already a biochemistry major, certainly it's a big helpful for that. If you never learned, it's still a good chance to learn here because we're going to talk a very basic material. And also this will have a lots of the extra points will be offered in the exams. So this one is a big one. And exam 4, what we're going to learn, we're going to talk about is applied microbiology. Applied microbiology, for example, we will talk about is food safety. So what is the food safety in the United States? Because my research is focused on the pathogen control during the food processing. That's applied microbiology. We're also not going to talk about the birds processes. So you have raw birds processed until the carcasses in the Walmarts you could get and to cooking. What's the procedure to make sure the safety? So that's applied microbiology. Something will be very specific to the West Virginia. So basically what we have here, identification plus numeration. That will be cover the whole classroom. Okay, so we talk about that. That's microbiology, the science of microbiology. This is, you can read the slides, concern the study of microorganisms including algae, bacteria, virus, protozoa, and fungi. We will be focused on bacteria here. We will mention the virus. The reason is that in the last couple of years, you have been heard about the virus all the time. You may feel tired about that, but bacteria, honestly, you haven't heard too much. So we're going to talk about the bacteria, focus on that. Okay, the next question, microorganism. Okay, microorganism. This word is very good. It's a magical word, I always say. We could split them into the two sections. What is micro? That is too small to see by naked eye. What is organism? Certainly this means live body. Okay, so it's too small to see by naked eyes. We have to use a tool. The tool is very obvious. It's called microscope. And in the lab section, we will be focused on the light microscope. And also we're going to focus on the bright field microscope. That's what we're going to stick onto the lab. Okay, live body, what are the examples? Let's just give you some of the examples. We have slides right here. That is tedious. Okay, everything is there. But we want to make sure you're understanding, so I usually draw on the blackboard to talk about that. We said this class will focus on the one microorganism is bacteria. So, you know bacteria. Okay, what are the examples for bacteria? Everybody knows E. coli is Escherichia coli. And I make the jokes all the time. I said, even if you failed AEM 341, you come out of the classroom, and 10 years later, maybe 20 years later, when you are getting older, you still remember one bacteria, which is E. coli. Everybody knows. So that's E. coli. And you have pretty much heard about salmonella. If you see the news, salmonella is always contaminated with chicken foods or eggs. And you may have heard about listeria, which is a pregnant woman will be a big risk for listeria monocytogenes because it will cause miscarriage. And it is not recommended to eat daily foods like hot dogs, like hams. So that's bacteria, the first one. Okay, the first category. Okay, let's talk about another category. It's at the bottom. Virus. Last couple of years, people don't want to talk about this, but microorganism, we have to talk about virus, okay? Which one we talk about? COVID-19. Is that right? Coronavirus ID 2019. That suffered for all of us for 3 years. And more than that, then we have a debate about whether you use a vaccine. And finally, I mean, everybody got it. And then we have lots of testing. Then finally, everybody have it. Then right now, nobody talk about that anymore. Okay, so that's one. Okay. Rabies. Remember that from the dogs, we also call it a crazy dog virus. And in the United States, we say in 2030, we will be concave the rabies. So that's a virus. Now, what's a virus looks like? Virus looks like very special, somehow like that. If we remember, is that right? Somehow like this. Then the middle will be the DNA or RNA, which is nucleic acids, never goes, and packaged with a protein. It's very simple. And we say coronavirus, it's a crown. What it looks like? It's like some crown like this. Is that right? Something like that. Looks beautiful, but harmful. And we get protein, we get the S protein, and then we have vaccine. Okay, we're going to talk about the vaccine very quick. Okay, so that's something about the virus. Okay, so let's go, I'll put number 2 here. Okay, number 3, let's go back to top. Everybody knows fungi, is that right? Yeah, everybody knows fungi. So fungi have 2 sections, yeast and mold. So that's a different cultivation temperature. Yeast usually, it's a little bit higher temperature, 37, 35 degrees Celsius. Molds, 25 degrees Celsius. So what are they? Where you can see it? You have a bread you buy from the Kruger, then you, for the breakfast, you bite it, and you left it in the dorm on the bench for 1 week. Then one day you suddenly want to eat it, but you cannot eat it because you see those green color there. That's molds. Okay? So that's fungi. Now let's go number 4. What else we have? Protozoan. In here, the slides, we call it protists. Okay? But protozoan, you know that the protozoan. Together with the protozoan, we have algae. We have slime modes. So protozoan, you know that. You need a host. It's a parasite. You need a host. It's a protozoan. So for example, you had Toxoplasma gondii. The pregnant woman is not recommended or suggested you have a cat at home because it will cause miscarriage. Algae. This one is usually an environmental microorganism because it contributes to 80% of the oxygen generated in the earth. And slime modes, I say this is a very weird and special protozoan because it has a two phase. It has a yeast phase, or we say it's a fungi phase, and it also has a protozoan phase. So it's a double phase. That's called slime modes. Okay. What else we have? Fungi, protist, bacteria, we have archaea. So number 5, we have archaea. Archaea, we gave you example, which is a mesogens. It will generate a meson. CH4. It will generate a meson. What is meson? It's very simple example. We will be doing some of the lab work. During the fermentation, we put a grape. The grape with sugar and fermented will generate wine. And the alcohol will be generated. And some of the students said you use grape to do the fermentation. Can I use apples and the others? I said don't do it because it will generate a meson. That will cause blind eyes. So don't do that. That's what the meson comes out. But in the environment, the meson comes from the archaea. Okay, so this is all the examples. These examples, we say it's a cellular. Because cell is a single unit. They're all going to be composed by a single cell. So that's why these we call it a cellular. But the virus is not a cellular because there's no single cell can represent that. Therefore, it comes out with other sections. So we have varioids. Varioids, that's a single AEM. This is the one usually is caused damage for the plants in Philippines in 1996. That's a varioids. I will say this is simplified from the virus. It's called varioids. Another one. This one, I never heard about that. It's in the slides, which is called a satellite. Okay? Satellite, I never heard about that. So what happens is, when you see the virus, you have a protein, you have a DNA and an RNA. So single RNA become varioids. It could be the nucleic acids or protein, they become a satellite. So that's a very special microorganism. Is that right? Compared of the nucleic acids or the RNA. Last one, which is prion. This is the one composed only by protein. Let's say BSE, bovine spongiform encephalitis. It's happened in 1997 in Great Britain. It's a prion which is contaminated with the brain of the sheep, and the sheep were showing lots of the damages. And it could be transferred to the humans. So the only thing what they can do is kill all the sheep and do the deep burying. And because of that, the United States and Great Britain have a big dispute about the meat exchange and the imports. And the BSE, this prion has been transferred, or we say contaminated, into the United States in 2003. So even today, let's say China, they still forbidden the beef products shipping from United States and Australia to them. So let's say my family is in China, and if I want to be shipping them a bag of the beef jerky, it will not passing the border cross, the border patrol, because they are not allowed. You have they think the beef jerky may be contaminated with the prions. Okay, so that's something I want to mention. Now all these things right here, this is not a single cellular, so that's we call it uncellular. Because cell is not a single unit. Okay, so this is something we used to explain acellular, cellular, what the microorganisms looks like. And this is not uncellular, it's called acellular. So we just make sure it's acellular. Okay. So that's a basic overview of what the microorganism talks about. We have some of the genetic tools. We had the ribosome RNA could be isolated from the microorganisms, then to test the genetic difference between that. The closer the genetic difference, which means the closer relatives of those microorganisms. So all the microorganisms could be differentiated into three major groups, which is very clear in these slides, is bacteria, archaea, and eukarya. And the bacteria we talk about is a true bacteria. Okay? So remember, we could separate them into three major groups. This is based on the ribosome RNA. And we will talk about the ribosome RNA, what it looks like, what it's about. So next, some slides talk about the examples of the microorganism. Well, whenever we talk about microorganism, we always think it's bad. Okay? Because it's really harmful. So E. coli cause diarrhea, urinary tract infection, salmonella, virus. E. coli is not that easy. We want to dig a little bit more about that. This is E. coli. Okay. Whenever you heard about E. coli, everybody remember the name, but it's a story behind it. It's not that simple. We have foodborne pathogen, E. coli 0157H7. That's a foodborne pathogen. Let's say ground beef. It's a zero tolerance for E. coli 0157H7 because it will cause hemorrhagic uremic syndrome. And the kids will be died if they eat undercooked hamburger contaminated with E. coli 0157H7. It's a pathogen. And we also have generic E. coli. And generic E. coli, this is coming from our fecal sample, but it's not really harmful. So generic E. coli, we usually will say, we could see the fecal contamination. So we say it is an indication bacteria. And we also have urological E. coli. Because E. coli is one of the major pathogens called UTI, called urinary tract infection. And woman is a major risk group. And regarding the UTI, in the lab section, we will do a practice of measuring E. coli isolation identification in our urine sample. So that's a urological E. coli. And we also have E. coli 9037. That's a beneficial bacteria because they use for the fermentation. So just want to let you know, even you see the E. coli does not mean it's the same thing. It's a different categories, have a different purposes, and the results will be completely different. Okay? Salmonella, we already know it's a food poisoning. We will mention in our exam 4 when we talk about the birds processing. Virus, coronavirus, flu, hepatitis, we already mentioned. So that's bad, really. We have some pictures showing you it's bad. Look at the fish, bacterial infection of the fish. That will cause vibrio, could be. Conjunctiveness of the eyes. This is I always tell the people. If you have a conjunctiveness of the eye, you have to be careful. It's not really the eye problem. It may be the sexual transmitted disease. So we'll talk about that later. Okay. Some of you study plant science. A very rotted tomato. Could be contaminated with ammonia, which is a plant pathogen. Last one, everybody knows, strep rot, caused by Streptococcus pyogenes. We will mention detail in exam 3. And strep rot is most of the excuse before coronavirus. You were sending me an email said I'm not going to come into your class because I tested strep rot in the morning. Okay, so that's a strep rot. Bacteria always bad, not necessary. Sometimes it's really good. Everybody likes the alcohol beverages. We have the wine, which is fermented from the yeast. Okay, wine yeast. We have a beer fermented with Saccharomyces cerevisiae. We're going to do the observation in the lab to the manufactured, already made Saccharomyces cerevisiae, the slides. And we will talk about the fermentation, about beer brewing process. Okay, antibiotics, penicillin, tetracycline. We have a section talk about that. It's a metabolic ingredients coming from one microorganism and killing or inhibit another microorganism. Insecticide is the same thing, killing insects. The bottom one is everybody like it. You like cheese, yogurt, salt cream. It's a dairy product. It's a fermented product. Which bacteria it is? Lactobacillus. It's really the beneficial bacteria will adjust to your intestinal gut area. And always people say the intestinal gut area right now is the center of your whole body. It's not the heart, not the brain, it's the intestinal gut area. So just want to tell you one thing. A bacteria, microorganism is not always bad. It's a beneficial use. Okay? So this is some introduction. Before we move to here, we want to emphasize something. How many microorganisms in the world can you guess? It is more than 10 to the power of 10 to 10 to the power of 30. Huge. You cannot even count. And how many microorganisms we really know? Around 5%. So keep in mind, if we have microbiology class, you may have a clinical microbiology class, you have an MIC 200 in a health science class, different classes. We have millions of microbiology textbook. How many bacteria we know? How many microorganisms we know? Around 5%. More than 95% microorganisms, we don't even know where they come and what are they, but they existing. And people even predict, like COVID-19, these type of emerging unknown pathogen, it could come out every 15 years in the future. So just want to let you know. Although we have that's why when the coronavirus comes out, we don't know how to do it. Although we have so many publications, we have research done by more than 150 years or more than 200 years, because we only know about 5%. But 95% we don't even know. And compared to this, it is huge. Okay? So just want to let you know something before we talk about it else. Okay, so that's very basic opening remarks. Gave you an overview what is microbiology, what is microorganism. Now we're going to go talk about a little bit of history. Okay, vaccination. Why I move this one at the top? Because a couple of years we talk about coronavirus vaccines, so we move vaccination at the beginning. Okay, vaccination. Okay, this word is very magical. It is coming from a Latin word, VACCI. What this means? VACCI coming from a Latin word which means cow. So what happened is, in the old time, in the Great Britain, people suffered from a very severe virus, which is called smallpox. This will be completely destroy your bone marrow system, which is your immune system, the basic resource and foundation of your immune system. Because millions of people, trillions of people died in the world. And the damage, I would tell you, is much more than coronavirus. This is because of all kinds of reasons people make it a big deal. Smallpox, much more people will be killed. Another one is Spanish flu, we will mention later. Those two are much more people killed by the time because of underdeveloped technology, medical technology by the time. So, Edward Jenner tried to do something. What he finds, most people are sufferers from the smallpox. But there is one group of the people who will be well survived, which is a woman working in dairy farm, which means they close with the cow. And they have a very mild disease, we call it cowpox. So what happened is, he got the arm, okay, I'll just draw arm, not very good drawing. The arm had the lean, which is a cowpox. And he taking out of the lean from the cowpox, from the dairy farm woman, and make kind of like chemicals. Then the chemicals, which is injected to this baby, which is in a Royal Society of England. And then he contaminated with this baby with smallpox, and this baby is survived. And then later on, people would love to memorize Edward Jenner. They named this method is vaccination. They gave this terminology called vaccination. And I want to say this, whether you believe or not, still the best way to control virus-caused disease is vaccination. This is the reason when you're born in the United States, you require to have an immune schedule. Let's say HIV, Haemophilus influenza, type B Haemophilus influenza. Okay, those bacteria. And you have a vaccine. You get the vaccine and you can be survived. For the virus, whenever you have a cold virus, you go to see a doctor, then they give you some of the medicine, maybe like a cough inhibitor or something, but you're still going to recover about a week. There's no magic to really curing the virus, although there are not some new technology comes out. And for the virus, we only can do the prevention, and the vaccination is the best way, still written on the textbook, is the best way to prevent or to protect it, or we can say it's a proactive way to prevent the virus contamination. Okay, so this is a vaccination. Edward Jenner is the first person in the world in the Royal Society to develop the vaccine. The vaccine is come from the cowpox, and he injected it into the small baby, and the baby contaminated with smallpox, and it is survived. So this is tells you, right there the note, it is preceded the work, established the role of microorganisms in the disease. So that's the first thing. Okay, microorganism. We talk about a microorganism. We say it is too small to see by naked eyes. So we need a microscope. At the beginning, it's not really light microscope. It is mechanic microscope. And the first mechanical microscope is generated at great Netherlands, or we say Netherlands amateur scientist, which is Antonio van Leeuwenhoek. Okay? Van Leeuwenhoek. He is an amateur scientist because he do the business of the trading in the European countries. In the amateur time, he likes to do some of the science. So he created this mechanic microorganism, a microscope. So you see there is a lens, there is a specimen holder, there is a focus, There's a handle, and you use your eye, you can do the observation. Now, we talk about a microscope, we always want to talk about is a magnification. We will be mentioning the lab real quick. Use light field microscope. The magnification is basically around 1,000x. And use Antonio van Leeuwenhoek's mechanical microscope. The magnification is pretty much about only 200x. But it is very important, it's a breakthrough, because he created a microscope, he observed, and very important, he described accurately and recorded. So you can see the picture number 3, which is what he recorded. What's a microorganism? He drew the observation by the time, use his own mechanical microscope. But later on, people find it's something interesting. When you see here, something like that, when you see something like this is not a microorganism cells. We will do the observation in the lab using light microscope. You will see this is not a bacteria. Where are they? Those are the flagellar movement of a bacteria. So, but we still credit the contribution him for the microbiology world. Something I also want to mention is this. You look at the time is 1632 to 1723. When we go to talk about Louis Pasteur, this is 1800. Okay, when we talk about Bob Koch, this is already 1900. There is about a gap, is 150 years as a gap. The science area for the microbiology is developed very slow. Why? Because Anthony van Leeuwenhoek is refused to sell his mechanical microscope to the world, to the other people. That's why it's delayed the development of the science for about 150 years. So he had some contribution for the world. People gave credits, but we also want to say there's something he should do better, okay, from the scientific area. Okay, next one. We want to talk about the spontaneous generation, something related to microorganism. We want to start from the not microorganism, from the real life body, spontaneous generation. We want to make sure it's relatively simple. I don't want to spend too much time stuck here. Spontaneous generation. So what means spontaneous generation? This means on live stuff, I'll just say, generate a live body. So said very simple. You have an empty vessel, a glass vessel. Then suddenly it's generate a mice. That's from a vessel to a mice. That's called spontaneous generation. Because of the political reason, because of the religious reason, the people who believe that for a long time. But the scientific area, they want to repel it. So Francisco Redi, a great Italian scientist, developed a very magic study, which is a raw meat and a maggot, to repel or discredit spontaneous generation from the live body, not in a microbiology level. Okay, so the story is that if I have a meat, okay, it's a raw meat. Okay, here is the thing we wanted, when we talk about this one, you have to understand is that there are two things here will bother you to understand the spontaneous generation. Number one is all the raw meat going to generate a fly. This will be a question. The second question, does the air will generate a fly? And you have to describe both. So it's very simple. You have a raw meat. Okay, you are exposed to the air. The fly comes out. That's very normal because the maggots comes out. But you know the fly lay down the maggots become a nymph, then finally become a fly. So what I do? I need to tell you the long meter is not generate a fly. The fly is coming from the maggot. So what I do? I'm going to cover with a piece of paper. Okay, so long meat. I cover with paper. Is that right? So, is there going to fly come out? No, it's not going to happen. That tells you long meat will not generate fly. But how do we know the air is not generate fly? This is really the beautiful thinking from Francisco Redi. He used this one. Gauze, or we say cheesecloth. What is the cheesecloth? It's exposed to the air, but the space between that is small enough, will not allow the fly to have a maggot. The maggots will not penetrate it. So when you cover with cheesecloth, is there anything happen? No. But when the cheesecloth removed, when you have this cheesecloth, there's no fly. Is that right? But when the cheesecloth removed, the fly gonna comes out. When this guy is removed. So it's a very magical experiment from a live body level to discredit the spontaneous generation. And the magic one is this cheesecloth cover. Okay, so that's very interesting. Now, when Francisco Redi comes out with the spontaneous generation, then there is come out with a debate about microorganisms. Okay, a very simple example. I will draw here. Okay, I have a bottle. Okay, a bottle, I'm going to put some of, let's say just put some of pond water. Okay, you get it from Cheat Lake, West Virginia, pond water. Then if this bottle, think about 200 years ago in the two labs, both of these people will do is boring, which means you heat it. They don't have an autoclave by the time, okay? They don't have a high pressure system. And then after boring about half an hour, 1 hour, when people find there is a bacteria survive, and when people find it's clear, nothing. Okay, then we'll have a debate. Where is the survived bacteria comes from? This is exactly John Needleman and Lazo Sperazzi find the opposite difference. John Needleman find is a mutton broth. You boiled, you sealed, it's become cloudy. Cloudy, which means turbidity, which means microorganism is growing there. We will do this in the lab. The simplest index of a broth media is contaminated or grow with bacteria it is become cloudy and turbidity. However, Lazzaro Sperazzi find it different. The same thing, he sealed, he boiled, no growth of the microorganism. So, who is correct? And when we learning in microbiology, we will be knowing this. Both of them are correct, both of them are possible. Why? First of all, they could generate heat resistant, they could exist in heat resistant bacteria. Which bacteria resistant to the heat? Where you can find it? In the hot spring or hot sprit in Yellowstone National Park. It's 120 degrees Celsius, 100 degrees Celsius. You boil in the whole day, the bacteria is still there. Heat-resistant bacteria. The second is, we talk about E. coli, we talk about salmonella. It's easy to kill. You heated it, it will die. But there's other bacteria, when you heat it, it does not mean they will die. Because they were fuming endospore. And it's an endospore forming bacteria. Let's say Clostridium, let's say Bacillus. That's why they will have opposite results, because it depends what are the microorganisms existing in the pond water. If you pond water have Clostridium, have Bacillus, you boil it, it may still become turbidity because they are resistant to the heat. But if you pond water only have E. coli and Salmonella, then it will become very clear because they are easier to be dyed when you're using heating methods. So this comes out with a very magic stuff is Louis Pasteur talk about swan neck flask. It's a very magic stuff. Okay, this one we just want to make it simple. If you read a textbook regarding the story of Louis Pasteur, there is a different version about this picture. But we will just make sure it's easy. Something like that. Okay. Okay, think about this. If I don't have this one neck, what happened? I have a pound of water inside. I have a pound of water in there. Okay, I heat it. It's killed everything. Become clear. Then I cut it. The bacteria from the air going to come in. It's going to become turbidity. When I have a swan neck, what happened? It's clear. Because the bacteria coming from the air, it's going to stuck here. It's not going to come. It's not going to go inside. What this tells you? This tells you if are sterilized, which means pre-treated pond water, in a swan neck, it will not get contaminated because the bacteria cannot penetrate, which means it will not generate a bacteria spontaneously from the pond water. Why the pond water will have a bacteria? Because the bacteria is coming from the air and it's contaminated. It's not spontaneously generated. Okay? So, this very magic swan neck bottle solved the problem. We will say basically 90% solved the problem in the world about spontaneous generation is not going to work even in a microbiology level. But people are still going to struggling with this question. How you explain it? And the people will repeat it. Using a same pound of water, use a swan neck, you heat it, they still find something survived. And finally is John Tidell and Ferdinand Cohen. This is two great German scientists. They spending lots of years to do the work. John Tidell find heat-resistant bacteria is existing. So some of the repeatments of Louis Pasteur's swan neck work does not repeat the same results because there is a heat-resistant bacteria. Another thing is I just mentioned from the Ferdinand Cohen, he found some of the bacteria they may have endospore will cause them heat resistant, like Bacillus and the Clostridium. Okay, so that is the reason why it still took a couple of years for the people to finally to discredit spontaneous generation in the microbiology level. This swan neck system tells you microbiology will not generate from sterilized pond water. And they're only coming from the air. However, if you repeat it, why you see a different results? Because they could exist in endospore forming bacteria or heat resistant bacteria. Okay, so let me see the time. Okay, so this is what we have for our first class, and Thursday we will be continuing to talk about Bob Cook and his